We just got done talking about how the contraction actually occurs, right? Through the cross-bridge cycle. Now, our skeletal muscles aren't going to contract all the time or stay contracted once they do contract. They have to relax at some point. So let's talk about the steps of skeletal muscle relaxation. And there are two major components. One is that the motor neuron just has to stop sending impulses. So the motor neuron action potentials stop. And that stops the signaling for the release of acetylcholine. Remember, the acetylcholine is the neurotransmitter which binds to those ligand-gated sodium channels on the motor end plate, causing an end plate potential. Well, if motor neurons stop sending impulses, then it's going to stop signaling for the release of acetylcholine. No more acetylcholine, no more motor end plate potential. All right, so those action potentials, not only on the motor neuron, will stop, but then that leads to the action potentials being sent along the sarcolemma to stop. All right, so the motor neuron has to stop signaling. It has to stop sending action potentials. No more release of acetylcholine. And then the second major part is that the calcium ions that have been released by the sarcoplasmic reticulum in the skeletal muscle cell, those calcium ions have to be actively pumped back into the SR. And that's because in order for muscle contraction to fully stop, we have to remove calcium from troponin so that tropomyosin can cover up those active actin binding sites. All right? So two major components. The motor neuron has to stop sending action potentials, no more acetylcholine, and then those calcium ions have to be removed from from within the cytoplasm of the skeletal muscle cell. We have to pump them back into the SR. All right, so if acetylcholine, once the motor neuron stops signaling, that means no more new acetylcholine will be released via exocytosis. But whatever acetylcholine remains in the synapse has to be degraded. And so remember, we talked about at the neuromuscular junction, I already mentioned an enzyme, a very important enzyme called acetylcholinesterase. And that degrades the remaining acetylcholine, which would be found in the synapse and could potentially lead to end plate potentials. So all that acetylcholine gets chewed up. No more acetylcholine means that the ligand-gated sodium channels on the motor end plate will close. That stops the end plate potential because no more sodium can enter into the skeletal muscle cell. That leads to repolarization. So if no more sodium is allowed to enter, then the membrane will repolarize back to resting membrane potential. Right, so the sarcolemma returns to resting membrane potential as the result of no more sodium is being allowed in because those ligand-gated sodium channels are closed. Potassium will leak out through those the leak channels, but then we also have you'll see that this will become. I'll talk more about this when we get to the nervous system, but there are voltage-gated potassium channels which open as well, and potassium will leave through those, helping to repolarize the membrane. Calcium channels in the sarcoplasmic reticulum will close as a result of no more action potentials being sent down the sarcolemma, right? So those voltage-gated calcium channels on the SR also close. All right, so the sarcolemma will return to resting membrane potential as a result of potassium leaving, no more sodium is allowed in, and then the calcium channels on the SR close. Calcium ions are pumped back into the sarcoplasmic reticulum. So pumped, that is a term we use when we are referring to active transport. So active transport means it requires ATP, and that's because these pumps are pumping calcium into the SR against its concentration gradient because there's more calcium in the SR than in the cytoplasm. And we're pumping more and more calcium out of the cytoplasm and into the SR so that contraction can stop. In the absence of calcium, so once all that calcium gets pumped back into the SR, in the absence of calcium, troponin and tropomyosin block the active sites on actin, and the muscle can relax because no more cross-bridge cycles. If tropomyosin slides back over and covers the active sites on actin, then the cross-bridge cycle cannot happen because those globular myosin heads no longer can attach. And then the myofilaments will slide back to their original positions, lengthening all of the sarcomeres, lengthening the myofibrils, and the skeletal muscle relaxes. And here is just a summary slide showing all of that. And so you can refer to this as a summary slide that shows you all of these steps, which is, we've talked about everything from the generation of action potentials along the motor neuron, what happens at this axon terminal, what happens at the motor end plate, what happens down the length of the sarcolemma, how calcium gets released. Once calcium does get released, how does the cross-bridge cycle happen? We went through all of those details, and it's all summarized here. Now for relaxation, I'm going to refer to this video so it can help you visualize it.
Before we proceed to examine muscle relaxation, let's quickly recap the events of muscle contraction. After the neuron stimulates the muscle at the neuromuscular junction, muscle excitation spreads through the sarcolemma and down the T-tubules and results in contraction. Relaxation occurs when contraction ends. Contraction ends when neural stimulation stops and the calcium ion concentration inside the muscle fiber returns to the resting level. Let's review the sequence of events that returns the muscle to rest. When neural stimulation stops, the axon terminal no longer releases acetylcholine. The acetylcholine remaining in the synaptic cleft is quickly degraded by an enzyme that resides there called acetylcholinesterase. As a result, the cation channels associated with acetylcholine receptors close, and the motor end plate repolarizes and returns to its resting potential.
All right, so the signals along the motor neurons stop, so no more acetylcholine will be released via exocytosis. But there is some acetylcholine that has already been released and remains in the synaptic cleft, or in the synapse, right? So acetylcholinesterase is an enzyme that's always present in the the neuromuscular junction. Acetylcholinesterase quickly degrades the remaining acetylcholine, and when there's no more acetylcholine, that means these ligand-gated sodium channels, which ligand meaning it requires acetylcholine bound for it to be open, these ligand-gated sodium channels will close, no more sodium is allowed to enter, and so the depolarization at this end plate stops. And it will return to resting membrane potential.
The repolarization spreads to the rest of the sarcolemma when voltage-gated sodium ion channels close and voltage-gated potassium ion channels open.
So it's switching back. So this video shows you that it's switching those charges, that charge distribution switches. So it started out being this sarcolemma was depolarized, right? Because we just had action potential being sent down the length of the sarcolemma. And when it's depolarized, remember the inside of the sarcolemma will be less negative or more positive. But we are repolarizing now because acetylcholine is no longer stimulating this end plate. So what happens is our voltage-gated sodium channels close, and then voltage-gated potassium channels open. And where is the gradient for potassium? Remember, there is more potassium inside than outside, right? So if there's more potassium inside than outside, once these voltage-gated potassium channels open, potassium rushes out. Well, potassium's a positively charged cation, and so when potassium rushes out through these voltage-gated potassium channels, as well as through leak channels, which aren't shown here, then it's going to make the inside of the sarcolemma return to its resting membrane potential, which is negative. So it will become more negative or less positive. Either way, okay? Just it returns it back to resting. And remember, resting membrane potential is around minus 85-ish millivolts.
This allows potassium ions to leak out of the muscle fiber. When repolarization reaches the T-tubules, calcium ion channels in the membrane of the sarcoplasmic reticulum close. The calcium ions remaining in the cytosol of the muscle fiber are quickly pumped back into the sarcoplasmic reticulum via active transport.
So when the membrane, the sarcolemma repolarizes, meaning it's back to resting membrane potential, well, when it returns to resting membrane potential, these voltage-gated calcium channels close because it's no longer being depolarized. It's resting. All right, so these voltage-gated calcium channels open, no more calcium can be released into the cytoplasm, and then whatever calcium is in the cytoplasm needs to be pumped back into the sarcoplasmic reticulum and stored there until the next time we want to do skeletal muscle contraction. And the way that we get calcium back into the sarcoplasmic reticulum is through these calcium pumps. Pumps, anytime you hear the term pump, you should immediately think it requires ATP energy. It is active transport because we're pumping calcium, more and more calcium, into the sarcoplasmic reticulum against its gradient. Why is it so important for calcium to be pumped back into the sarcoplasmic reticulum and stored there until next time we want to do contraction? Is because if calcium just stays in the cytoplasm, then the skeletal muscle cannot relax. It will stay contracted. So both we saw in the cross-bridge cycle, which is active contraction, right? The cross-bridge cycle requires ATP. And then skeletal muscle relaxation, the pumping of calcium back into the SR for relaxation, also requires ATP. So contraction and relaxation requires ATP.
As you have previously learned, active transport requires ATP. Recall that ATP is also used in the cross-bridge cycle to cock myosin heads. You can now see that ATP is important for both contraction and relaxation of muscle. During the final stage of relaxation, the cytosolic concentration of calcium ions decreases and calcium ions let go of troponin. As a result, tropomyosin slides back to block the active sites of actin. The muscle contraction is now over. Eventually, individual sarcomeres and the entire muscle will return to their resting length due to external forces such as tendon recoil, contraction of opposing muscles, and gravity.
All right. All right, so now let's do some review questions. During muscle fiber relaxation, calcium channels in the SR close because... All right, so how do those calcium channels in the SR close during relaxation? Okay, the resting membrane potential is restored. Calcium levels in the SR are depleted. Calcium is released from the SR, or calcium is pumped into the extracellular fluid. Why do those remember, those calcium channels in the SR are voltage-gated calcium channels. They open when the sarcolemma depolarizes. Well, during relaxation, the resting membrane potential is restored, and no more depolarization means the calcium channels close. So that's the answer. The resting membrane potential is restored, therefore closing the voltage-gated calcium channels. During muscle fiber relaxation, calcium levels in the sarcoplasm, which is just the cytoplasm of a skeletal muscle cell, calcium levels in the sarcoplasm rise? No, that's incorrect because we know calcium levels rise during contraction. Calcium is pumped back into the SR. Okay, let's keep reading just to see the other choices. Calcium is released from the SR. No, that happens during contraction, or that's the link between excitation and contraction, is the release of calcium. Calcium is pumped into the extracellular fluid. No, that's incorrect. Calcium is going to be pumped back into the SR so that it can be stored and used again the next time that skeletal muscle has to contract. All right, so calcium is pumped back into the SR. Acetylcholinesterase is in the synaptic cleft, degrades acetylcholine, allowing depolarization of the motor end plate. Well, we know that's incorrect because it's acetylcholine binding to those ligand-gated sodium channels that depolarizes the motor end plate. So if acetylcholinesterase is degrading acetylcholine, then it's not going to allow for depolarization because it's degrading or getting rid of acetylcholine. Calcium levels in the sarcoplasm rise. That's incorrect because it's when acetylcholine is bound to the ligand-gated sodium channels that causes depolarization, which leads to an action potential, which leads to calcium being released from the SR. Tropomyosin, so acetylcholinesterase in the synaptic cleft degrades acetylcholine, allowing tropomyosin to expose the actin active sites. Well, that's incorrect because that happens when we're having muscle contraction. Ligand-gated sodium channels to close. Yeah, that's the correct answer. So acetylcholinesterase in the synaptic cleft degrades acetylcholine, allowing ligand-gated sodium channels to close, because if we do not have acetylcholine is what binds to those ligand-gated sodium channels and opens them. If acetylcholine is degraded, then those ligand-gated sodium channels close. Sarcolemma repolarization during relaxation means that the interior of the cell becomes less negative. Well, remember depolarization, right? During excitation, depolarization is when the inside of that plasma membrane becomes less negative or more positive. Less negative, meaning moving in a positive direction, less negative. So that's incorrect. Restores the resting membrane potential, okay? Only occurs at the motor end plate of the fiber. No, we saw in that video that repolarization happens down the entire length of the sarcolemma. Is caused by the closure of the calcium channels? It restores the resting membrane potential. So the sarcolemma repolarization during relaxation restores resting membrane potential throughout the sarcolemma, throughout the length of the sarcolemma. And this is just a picture back from the previous slides that when we talked about how do we generate resting membrane potential and action potentials, just so that you can again see this. Resting membrane potential is again measured just along the inside of the sarcolemma or any plasma membrane. It's going to be about minus 85 millivolts. Now, when sodium channels, like ligand-gated sodium channels, are open, sodium rushes in because it's going to follow both its concentration and its electrical gradient. When sodium rushes in, that's called depolarization. And the membrane is becoming, less negative, more positive. It's moving in a positive direction. All right. Now, during repolarization, those sodium channels are going to close. No more sodium can enter. And potassium channels open. And these voltage-gated potassium channels, when they're open, potassium rushes out. And when potassium, which are positively charged ions, rush out of the cell, the inside, where we're measuring membrane potential, is going to become more negative. So it's moving in a negative direction back to resting membrane potential. So that's what repolarization is, moving in a negative direction. So if it helps, draw this out so that you understand and make sure you label your axes from negative to 0 to a positive value up top so that you understand that depolarization is due to sodium channels being open. The membrane becomes less negative, more positive. Repolarization is due to potassium channels open, potassium leaving the cell, and it becoming more negative, moving in a negative direction back towards resting membrane potential. Which aspect of muscle relaxation requires ATP? Okay, motor end plate repolarization, blockage of the actin sites by tropomyosin, sarcomeres returning to their original length, pumping calcium ions back to the SR. So which aspect of muscle relaxation requires ATP? Pumping calcium ions back into the SR. Remember, those calcium pumps are pumps. You should always think active transport proteins. It requires ATP. So both muscle relaxation and muscle contraction requires ATP. Those calcium pumps that pump calcium back into the sarcoplasmic reticulum to be stored for later requires ATP. And that's where now we'll talk about rigor mortis. So if you've ever heard, rigor mortis it literally means like stiff death. So when an individual dies and 3 to 4 hours later, they become really, really stiff. The reason for that is, all right, so the progressive stiffening or the contraction of skeletal muscles upon death, or 3 to 4 hours after death, is because of the fact that those calcium pumps that pump calcium back into the sarcoplasmic reticulum no longer have ATP to fuel their activity. So whatever calcium has been dumped into the cytoplasm can no longer be pumped back into the SR. So calcium remains in the cytosol, bound to troponin, and then those cross bridges can start. And then the fact that, okay, so upon death, your mitochondria are they're no longer making ATP. So calcium can't be pumped back into the SR, but also remember in the cross-bridge cycle that ATP is required for the myosin heads to detach. Remember? So ATP has to bind to the myosin head for it to detach. Well, your mitochondria are no longer making ATP because you're dead. So calcium is still present, bound to troponin, and there's no more ATP to detach those myosin heads, so that upon that very last cross-bridge cycle, it just stays contracted. There's no relaxation because ATP is needed for relaxation. Okay, so muscle fibers are unable to relax without ATP, and so calcium can't be pumped back into the SR, the myosin heads can't detach from actin, and so those muscles remain contracted until a long enough time has passed to where the proteins start to degrade. About 48 to 72 hours after death, those proteins start to just completely degrade, and then that dead body would relax. Okay? So if you've ever heard the term of the term rigor mortis, now you understand, with all the muscle physiology you just learned, why there would be stiffening or muscle contraction once someone has died. And this is just the big picture. This is a nice summary slide that you can look through and read through all of the steps of excitation, contraction coupling, the actual contraction, and then relaxation, which we've gone through in the last several videos.